Chapter 53, we're going to be looking at population ecology now. So in Chapter 52, we looked at an introduction to biology, and we learned that ecology could be studied at a variety of levels. The largest level being the global ecology, which is where we look at the entire biosphere, abiotic, biotic factors. And then the next level down underneath global would be landscape ecology, followed by ecosystem ecology, then community ecology, and population ecology came underneath that. So if you recall that tiered image that we looked at, which is found on page 1145, you'll see what I'm talking about. So we're going to one of the smallest levels of ecology, talking about populations. Chapter 54, we go to the next step above population. We'll be looking at community interactions. So moving on with population ecology. We're going to be looking at populations and population density, dispersion, things like that. So let's start with population, just to recap what it is. I know we already spoke about this one. It's a group of organisms of the same species that are able to reproduce and produce viable offspring and live in the same area. Populations are described by their boundaries, where they happen to inhabit, which area they live in, as well as the size of the population. Now our two words of focus for the next few slides here are going to be density and dispersion. Density would refer to how many individuals of a species are living in a defined area. For example, how many students are in my classroom? That would be the density of my classroom. That would be a defined area. Another example would be number of oak trees per square kilometer in any county. Dispersion would be the spacing between individuals. Are the individuals clumped together? Are they spaced out evenly? What's the dispersion? Determining density. Density is dynamic, so it is changing. In very, very, very rare cases, a population can actually be counted. So we can go and we can count how many individuals in a population there actually are. 1, 2, 3, 4, count. However, that is tough to do in most circumstances. Why is that tough to do? The area might be too huge. Could you imagine going from tree to tree in a large forest, counting how many oak trees are there? That'd be a lot of time. You also have some organisms, like cheetahs. They immigrate or emigrate. They move around. It's really tough to count organisms when they're moving around on you. And lastly, what's the birth rate and what's the death rate? Those change. So they are dynamic. Makes it difficult to actually go through and count. Because of these factors, most often, the density of a population is estimated. One method of estimating would be doing what is called the mark recapture method. This works great for individuals that can move about, such as animals. And basically how it works is you'll physically catch some organisms, and you'll put a tag on them. And then you'll release them back to the wild. And then you catch more of those organisms. And when you catch them a second time, you see of those that you caught, how many have the tag from the first time and how many do not. And you can take those countings and plug them into an estimation formula, and you can figure it out. This works even with sharks, who you can't actually catch, but you have to kind of just tag them with a poking prodder. You very commonly will see ecologists do this in the field with butterflies as well as crickets, in which case you can sweep through a field with a net and catch those crickets, which is a great effective way to understand how the mark recapture method works. So patterns of dispersion. There's three we're going to be looking at. The first is clumped, which is kind of what we see going on in this image here. We have a defined area, and we see small little clusters of organisms thriving together. That would be clumped dispersion. Organisms that might do this would include plants and fungi, and they'll clump where soil conditions or other environmental factors favor the germination of the seeds and the growth. So there's something in the soil here perhaps that favors a small patch to grow, but right here that condition is not present. The next method of dispersion is uniform, which is what we see going on in this image here, and we can kind of see that going on with the penguins as well. This happens in both plants and animals, where we have the uniform distribution. Everyone has their own personal space. This happens in plants because some plants will put off a chemical that will prevent other things from growing around it. Pine trees kind of do this because they make soil acidic, so a pine tree will grow and it's very acidic around that pine tree, so no other plants can grow. Animals do this through exhibiting territoriality, where they kind of claim their own space and they'll bump heads with anyone that tries to invade their personal bubble. And then the third is random, where there's just a random scatter of organisms, and this can occur when there's strong attractions or repulsions between certain factors. So it is, as you can see, quite random. We can see the randomness of these dandelions in this particular field, which is a result of just random wind where it blew the pollen. So looking at the demographics of a population, to start with demographics, demography is the study of vital statistics of populations and how those populations change over time. And a few things we're going to look at that are used to study demography include life tables, survivorship curves, and reproductive rates. So let's take a look at life tables. Life tables are used to show in a cohort of organisms how many are alive at each age. So age 0 to 1, age 1 to 2, 2 to 3, 3 to 4, so on and so forth. Of the initial cohort of 337, all of them are alive. 100% of them are alive. But after the age of 1, moving into the next age bracket, during this year, in this case, 207 happen to have died. So how many are alive at the start of the second year? About 38.6% of them. So 252 are alive from the initial, and we lost 125 moving into the next one. So as you can see, the number of the cohort slowly decreases over the course of time, as does the number of deaths conversely. It's actually decreasing because there's less individuals to be killed off. So we can look at demographics in one fashion, looking at age-specific summaries, and we'll look at a different form of a life table when we look at human populations near the end of this chapter. So our next one is survivorship curves. Survivorship curves are graphed over the number of survivors over a lifespan. And there's three generalized types of survivorship curves. Humans apply to the first type of survivorship curve, or survivorship curve Roman numeral 1. What this says is that humans tend to have a long lifespan, as indicated by most people living past age 50. But what happens to people after age 50? They slowly start to die off. And how many people are alive at age 100? Not many. Other organisms exhibit a survivorship curve type 2. And this is where organisms slowly but consistently die over the course of the years to produce a straight line that gradually decreases to 0 organisms alive. And I guess this should be, this isn't 100 years old, this is 100%. My bad for talking about humans. And the last one is where there's a type 3 survivorship curve, and this applies to like sea turtles, for example, where there's a ton of them born and most of them die right away. And then the ones that do not die right away, they'll survive and they'll live for a very long time. How many will survive for a very long time? Not many, because there is overproduction and most died right away. So there's three types of survivorship curves: type 1, 2, and 3. 1, born, tend to stay alive for a long period of time. Type 2, born, Population decreases steadily and readily over the course of time. Type 3, born, most die. The ones that survive tend to live out the full lifespan. And the third one is a reproductive table, which more or less just indicates at what ages fertility happens to occur. Looking at this chart here, we can see the age brackets. Looks like we're looking at Belding's ground squirrels. And we can see what proportion of females in each age bracket produces a litter. And we can see at 0 to 1, right, that first year of life, no females are capable of producing a litter. They're not sexually mature. At the second age bracket, that second year of life, a little bit more than half are capable of producing a litter and do. The third year of life, a little bit more. Fourth year of life, more. Fifth life, a little bit more. And then the last half, all females are capable of reproducing. They've reached sexual maturity. Now what we're looking at in the third column is the mean size of a litter. So looking at this, it looks like the highest one is at roughly 5.5 squirrels per litter. So this would indicate that females are most fertile at this age range. This is when they produce the largest size litters. Notice that would produce almost a bell curve type graph if we were to graph these out, where most females produce their largest litters right in this range right in here. Can they produce litters younger than age 3? Yes, but they're smaller litters than at age 4 to 5. And also on the other end of that, can females produce litters when they're older than 6? Yes, they're just smaller litters than right at the peak right here. So this is what reproductive table shows us. It shows us the sexual maturity range of females and litter sizes. We'll pick up with the next concept in our second video.